Welcome to Based Theory. What you're about to see is an older debate of mine from around 2 years ago when I was just getting started before I had a channel against a creationist apologist named Donny, who runs the YouTube channel Standing for Truth Ministries, where they push young earth creationism. And Donny says he likes to focus on creationist genetics as the primary vehicle of what he thinks disproves the old earth, the scientific consensus, and the theory of evolution. So in this conversation, we have a very wide-ranging discussion on topics within genetics and creationism and the evolution debate. Specifically, we spent a long time talking about mutation rates, pedigree versus substitute, sex chromosomes, created heterozygosity, and finally we finish out by talking about genetic entropy and junk DNA. Like I said, this is from before I had my own channel, and it's when I'm relatively new debating against creationists. And my biggest regret is that I didn't cut Donny off more. Unfortunately, for those of you having to listen, Donny talks a lot more than I do in this debate. And there are certain times where it's just minutes and minutes of Donny monologuing. He's filibustering, gish galloping, doing it all, bringing up multiple topics, and it's just a lot to keep track of, and I apologize for anyone that I am subjecting to listen to Donny rambling down that entire time. But if you can forgive me, there's a lot of really interesting and entertaining parts of this debate as well. So let's get into it.
What would make sense both theologically and scientifically is that God would have created Adam and Eve heterozygous. So here on the left, scenario one, we see DNA copies are different. That's a heterozygous state, and that makes sense, okay? In my last 3 minutes, I want to briefly, at least, I'll probably only have time to cover one of these trees, and then in our discussion, I'll go over the second tree. So this is a tree specifically of all the major Y chromosome families in the world. And this was created specifically by Dr. Robert Carter, okay? There's history on this tree, guys. And looking at this tree, what do we see? Well, we see an explosion. We see a starburst pattern. Okay, we also see this in the mitochondrial tree, is this starburst pattern. And these branches, they reflect random mutations in people over time. Okay, Noah would be somewhere in the middle here of the tree. This is only a few hundred generations worth of time. It's incredibly easy to fit this number of mutations with a fast mutation rate in just 4,500 years. The amount of genetic variation that we see on the Y chromosome can easily fit within the biblical timescale. And this is perfectly consistent with separate ancestry. As a matter of fact, we couldn't ask for better evidence for separate ancestry.
Okay, so for starters, I want to say that last chart, it only has that starburst radial pattern because it's an unrooted tree. I mean, if you were going to look at this in regards to a molecular clock, you need to root that tree. It would look totally different. The length of lines there is representative of the time involved, but neither here nor there. I think molecular clocks is a good place to start this conversation.
Well,
and
real
also,
quick, Grayson.
Yeah.
What I'm going to do is I'm writing down your points.
Yeah.
So obviously we don't want any slapping and running. So take your time. So that's point one. What I'll do is I'll address them all when you're done.
Yeah. Okay.
Rather than interrupting you. Okay, so that's one. So I'll make sure I'm going to address that after.
I didn't know that we were going to be talking about created heterozygosity, but my only question there is where do you get the alleles if there's more than 4 alleles for a given trait, right? With heterozygosity, everybody has 2 alleles per person. So Adam and Eve, that's 4 alleles. So there are lots of human traits where right now in the population, there are more than 4 alleles for this trait. That could not be the result of created heterozygosity, right? So those are my first 2 points.
Okay, I appreciate it, Grayson. So let me share screen again on the phylogenetic tree. So you said that this is an unrooted tree and it needs to be rooted. See, an unrooted tree, this is an unrooted tree, but an unrooted tree in this context, especially as it applies to genetic variation, in this specific case, the Y chromosome. This is the mitochondrial DNA. An unrooted tree gives us the most natural reading of the data because the evolutionists, they have an assumption that, the last common ancestor or in terms of their, ape to man ancestry is at a certain spot. And then we, as biblical creationists, we can have our own assumptions. But if we manifest DNA variation in the form of an unrooted tree, what we get is a natural reflection of the data. And so therefore we can look at the patterns without having either the evolutionist or the creationist assumptions built in. Okay. So the branches
there.
Go ahead.
Okay, so if you're going to look at this tree and you're just going to make the intuitive assumption based on the data, you would think that the ancestor is at the middle of that starburst pattern. But what this tree is actually showing is the ancestor is further out along that longest branch. And so, like, if you were going to arrange this like a family tree, like how most people would think about this stuff, the oldest ancestor is not the center of that starburst pattern. That's not like once you root this tree and it looks like a family tree like everyone's familiar with, that's not where the ancestor is at. That's why this is not intuitive.
Okay, well, I'll address that too. I'll address them in turn. Okay, so again, what we have here in terms of the pattern, in terms of the number of DNA differences, okay? Because firstly, the branches in the tree, Grayson, they have mutations that we can count those up, okay? This is DNA data that you can get, and you can represent this tree yourself. You can do this yourself. And we know which mutation led to each branch, as a matter of fact, okay? So the point is the pattern, the variation, the number of mutations is easy to explain in the biblical time frame. There's not thousands of mutations here in the tree or in the mitochondrial DNA tree. We're only talking about a few DNA differences, okay, in both the mitochondrial DNA and Y chromosome DNA compartments, and therefore only a few hundred generations. Now, why it's important, the starburst pattern, okay, that you're contesting, is essentially because what we have is evidence for a rapidly growing population, okay? Accelerated population growth, essentially. And because it's an explosion from the center point, we may not know exactly where the ancestor is, but because this is evidence for a rapidly growing population, the ancestor therefore has to be somewhere in the center, because you have to have a starting point for where the population is now expanding from. Okay, so that's the point. But I would argue, and then I'll let you, actually, I want to answer your heterozygosity argument. I would argue it's even more difficult to place a point, a direct point in terms of where and who the ancestor is because of the branches themselves. So Grayson, if you were to look, let's say, let's use the mitochondrial DNA. If you were to look at
you can put it at one point. If you just root the tree, the same data, you can easily see the point that would be the root.
Right, but you're rooting it based on an evolutionary assumption. Okay, I would be rooting it based on a creationist assumption. You wouldn't therefore be getting a natural reading of the data. And why? Here's my main argument why I believe it's erroneous to root it as an evolutionist, especially, is because of the differing size branches. Okay, so take the most common haplogroup in Europe right here, HVR. Okay, this is the mitochondrial DNA tree for the audience sake, not the Y chromosome. Well, here's the thing. These people, they go back to a common female ancestor. And if all these people represented by these branches come from a common ancestor in the past, which they do, because this is just a natural representation of the DNA variation in the mitochondrial chromosome, okay, and the branch lengths equal the number of mutations, which we know is a fact, there's no dispute there, then that means in the same amount of time, the same family have picked up twice as many mutations as their closest living relatives. You have differing size branches. You have people living together, people living at the same time that are picking up, some of them more than 2 times the amount of mutations than their closest living relatives. So that means you can't look at the branches and determine where exactly that root is. You also can't make evolutionary assumptions in terms of when we apparently split from a common ancestor with the chimpanzee. You can't make those types of assumptions and determinations because of the differing sized branches. That means mutation rates are variable. Mutation rates are variable among living people and people groups. Before I go on, I don't want to say too many points. I've still got your heterozygosity objection noted, but I'll address that after. You can take some time to address what I said there.
Yeah, okay. There's not really much evidence that different types of people, like within each of these like groups, like we would expect, if what you're saying is accurate, that people of the L0 haplogroup, the longest line on this chart here, we would expect to find experimentally that their mitochondria have a faster mutation rate than people of the other haplogroups, and that's not what we see. So,
okay, so I would say you're just wrong on that, because firstly here, especially for the audience sake, I know you're familiar with this, I talk about it in my book here, estimates of the mutation rate with a non-phylogenetic approach, right, the pedigree analysis that I was talking about earlier, is approximately 10-fold higher than phylogenetically derived rates. So if you were to look to the evolutionary phylogenetic method rather than the pedigree method, they are admitting here in the secular literature, this is 2022, that your pedigree rates, your observed rates, are at least 10 times higher than phylogenetically derived
I
rates.
agree with that.
Right. But here's the problem, is where you're saying that we would need evidence for faster mutation rates. Firstly, the DNA data doesn't lie. We can see that with the branches. These people, these females here specifically, they go back to a common ancestor. Okay, you can see that with the branches where they go back to. That's the whole the point of the branching pattern. Okay, you have differing size branches, which means you have people who have more mutations, picking up more mutations than their cousins in the same amount of time. That's what these branches indicate. Okay, that's the evidence right there. But we have papers here. This is one from 2022. Notice this. 2 families had genetic drivers of germline hypermutation, with fathers carrying damaging genetic variation in DNA repair genes. So we have real-time evidence. We have empirical evidence. This is 2022, where we have certain peoples that are hypermutating. In this specific case, it's because they have deleterious mutations, damaging mutations in important DNA repair genes. That means you're going to accumulate more mutations because these DNA repair enzymes essentially work to fix mutations as they come in. Well, if these are broken, then obviously more mutations are going to get through, more mutations are going to be passed on in the germ cell line, okay?
But the point is that you have to demonstrate that these faster mutation rates are specific to these mitochondrial haplogroups, which is not demonstrated. I'm not arguing that families or individuals cannot be hypermutating and that mutation rates can vary on individuals and these kinds of things. What I'm saying is that mutation rates are not shown to be variable between different haplogroups for mitochondria or Y chromosome haplogroups. That is something that we just don't see demonstrated in the data. I mean,
well, Grayson, are you disputing the fact that we have, here's just one haplogroup in Europe, the HVR. Are you disputing the fact that we have differing size branches? The branches are reflecting what?
Time.
Mutations.
No, they're reflecting time. And here's what I'll say. You can do this. This can be empirically measured. You can find people in all over the world that have these different haplogroups, and we can look at their pedigree at a generation to generation mutation rate in their mitochondria, and we'll see that it's not dependent on their haplogroup.
Okay, so you're assuming that the branch lengths are an indicator of exact time. Now, of course, the more we move away to the center, yes, it is indicative of time. But we know that if you can pick up more mutations in the same amount of time, then you can't make an exact determination in terms of the branch length and correlate that to a specific time. What we see overall in terms of the pattern, but I did want to go to this paper here. Notice this. We also found that the overall point mutation rate is significantly higher. Notice this, Grayson. Significantly higher, and I'll even do it in italics, in Africans than in Europeans in this specific region studied, HBB. Okay. Over
in
here,
that specific region.
Well, of course, yeah. Some regions within the genome are going to be more mutable than others. But in this specific region, because we've made predictions on people groups whose mutation rates are not yet known. For example, as you know, Dr. Nathaniel Jeanson, he looked to the Khoisan peoples. Their mutation rate is not yet known. Okay. So he made a future testable prediction based on what we're talking about here, based on the data. And he said, I predict that they're going to mutate, this many times faster than some of your non-African people groups. That's a testable prediction on paper. So once we actually get their DNA and we can test their mutation rate, we can either prove that testable prediction to be false or true. Now, according to what you're saying, would you be willing to make a testable prediction based on the mutation rate in people groups who we've yet to measure? Would you be willing to, make a prediction like that?
Yeah. I mean, we can measure mitochondrial mutation rates for any people group. We're going to get same ballpark. It's not like the L naught in your prediction, the L0 haplogroup has to have the highest mutation rate in the mitochondria. That would be the prediction of the creationist model.
But again, mutation rates are not, now I would say they're constantly fast. We have a fast mutation rate in the Y chromosome. For sake of argument, we'll say 1. But what's up for debate is it's more like 3 when we look at high sequencing data. Okay. In the mitochondrial DNA, for sake of argument, we could say 0.1 per generation. It actually looks like it's as high as 0.5, but nonetheless still high in both DNA compartments. Now, if we have, and we'll keep rocking this topic till the 15-minute mark, but if we have low variation in both the mitochondrial phylogeny, Y chromosome phylogeny, okay, low variation, there's only a few differences. For example, here's a paper here. I wonder if you've read it. Right here, mitochondrial diversity within modern human populations. So this was published in a secular journal, Dr. Rob Carter and Dr. John Sanford.
So here Donny brings up a paper by the creationist Rob Carter and ghostwritten by John Sanford, another creationist proponent of genetic entropy. And this paper really didn't have that much to do with what we were talking about. It's all about the mitochondrial Eve consensus sequence, this total dream fantasy that creationists have, that they think that they could somehow just use the majority of alleles in the population today to somehow reconstruct the genetic sequence of Eve back in the biblical days. So yeah, it's just a complete gish gallop filibuster. It didn't really have anything to do with what we were talking about, and he talks about it for a while. So out of consideration to you all, I skipped that portion and then went ahead to where I tried to rope the conversation back into what was relevant for what we were talking about. Okay, well, something does need to be addressed. First of all, there is not any proof that people can have variable mitochondrial mutation rates. That hasn't been demonstrated.
It's right there in the tree. I feel like you're just repeating talking points.
Again. Like that's not measuring the tree. That's measuring the differences, right?
Yeah, but it's represented on an unrooted phylogenetic tree.
Exactly.
And so the branches are, but wait, the branches are allowed to naturally spread out. All this is
the center of that tree is not where it's.
We're not talking about the center. We're just talking about your different haplogroups and the fact that within those haplogroups, you have differing size branches. Well, if the branches reflect mutations over time, then that means if you have a longer branch and then a couple shorter branches in the same amount of time, that means you have people picking up more mutations than other people in the same amount of time.
Yeah,
that means
mutation rates are variable. You're starting from, right? If you start from the middle, then one branch seems longer than the other. If you start from a different area, all the branches are the same length, and the mitochondrial mutation rate is the same for all branches. It depends on where you start. That's why this graphic is worthless, because it's not rooted.
Well, it's not worthless.
So, anyways, the main thing that I wanted to move to was you're using the generation to generation pedigree mutation rate rather than using the substitution rate that's empirically found out.
Well, okay? Hey, Grayson, before you move on, I feel like you didn't answer the question. First, okay, we're just going to have to agree to disagree, I guess. Whether it's rooted or unrooted, it's still a representation of worldwide mitochondrial DNA. Okay, in that paper by Dr. Rob Carter and Dr. John Sanford, published in a secular journal, where they discovered the Eve consensus sequence, which again, as I explained, is not difficult to find at all. What did they find? They found that the mutation rate is fast, and they found that on average, we differ by the Eve consensus sequence by only 20 to 25 mutations. That's incredibly low. And so that fits perfectly within the biblical timescale. But you want to take this tree, the variation present in this tree, whether it's rooted or unrooted, it's the same variation. Okay. How do you take that back 200,000 years? That's a long time for such low variation. Okay, go. Are you saying because the pedigree versus?
Yeah, because your mutation rate is way faster than the rate that you should be using, because that you can't use the per generation like mutation rate, like how many mutations occur in me versus my parents versus my grandparents. That rate cannot be used, because 99.99% of those mutations are going to be lost due to genetic drift. So it doesn't make sense, right? You're saying that there are these 21 differences or whatever, but you can't use the per generation rate to calculate how many generations since there were no differences, because you can easily go back in time. Like ancient DNA is an entire field of science emerging in the early 2010s. We have ancient DNA. We have ancient mitochondrial DNA. We can go back and look at them, and it's not like they fit these predictions. The amount of differences between our mitochondrial DNA and the mitochondrial DNA of ancient DNA that we've found has, you would expect it to have somewhere between the different, like somewhere less than the 21 differences, right? But that's not what we observe. Like the number of differences predicted by the generational rate versus the amount of differences that we actually observe, like when we dig these people out of the ground and get their DNA, does not match. It matches the substitution rate.
Okay, so a few points there. Again, if the mutation rates are variable, and we can see here, okay, 2022, 2 families had genetic drivers of germline hypermutation. We can see this today through genetic sequencing studies. Okay, and what they conclude is that environmental exposures, including ionizing radiation, can actually influence. So there's a number of influencers in terms of the mutation rate, how many mutations are transmitted to offspring. Okay, so we have real-time evidence for hypermutation. We also have real-time evidence here. Okay, this is another paper from 2022. We have specific regions within the genome where it's significantly higher in Africans than in Europeans. Okay, so you have to say, and correct me if I'm wrong, your phylogenetic rate is more accurate, because essentially the reason why we have such low variation, few DNA differences in the mitochondrial chromosome, is because although the per generation mutation rate is fast, okay, most of those are going to be lost over time and are never actually fixed. So fixed just means get stuck in place. And so it's the fixation rate or substitution rate that we should be looking at. And you're arguing, and I just want to represent your argument accurately, you're arguing that the fixation rate or substitution rate, that's a lot slower than the observed mutation rate.
Yeah, because 99.9% of the mutations that occur from each generation to the next generation are going to be lost very quickly. When we actually are measuring the differences between two groups, the genetic differences that we're measuring are the genetic substitutions. That's why we have to use the substitution rate, because we're measuring the differences in the substitutions.
Okay, so here's my response. Okay, a few problems is your substitution rate is making a couple assumptions. Okay, firstly, it mostly assumes, I'm saying mostly because I know there are a couple examples of islands like the Canary Islands where they've done mutation rate tests, okay, to basically test the observed mutation rate with the long-term substitution rate or the accumulation rate. Okay, but most of the times that you read these papers, even in the Canary paper that Dr. Danster and Cardinal likes to cite, they admit 5 or 6 times that they are assuming humans and chimpanzees did split from a common ancestor about 7 million years ago. Okay, so in order to get your long-term substitution rate, your long-term accumulation rate, is you have to look at the DNA differences that, let's say, separate humans and chimpanzees Okay. Assume that we split from a common ancestor roughly 7 million years ago. Okay. And then considering those DNA differences that separate us, well, now you're going to have a really slow accumulation rate.
We don't have to do that.
I know you're going to look to the Canary Islands paper, and there's been a few other.
You can look to any, even within like the last couple hundred years, or you can calibrate your substitution rate based on whatever data you want. You can do it.
That's the key word, though. Calibrate.
Yeah, so because that's how you're getting the rate. Is you have to look at an ancient like DNA. This could be ancient as in like 200 years ago, like no.
Yeah, but you're okay. So if it's 200 years ago, you're calibrating with what? Archaeology.
No, you could. Okay, look. We agree when the Roman Empire existed, right?
Right.
You can go to an archaeological site of a Roman, like a Roman archaeological site. We dig up, we find a Roman, we test their DNA. We
so
look
2,000 years ago?
Yeah, like 2,000 years ago, right? This is well within the creationist time frame. We look at how many differences are between that Roman and then two of its ancestors from the modern day. And using that, we can calibrate our substitution rate. That makes a prediction, and then we can go dig up another.
Where did they do this? And where did they do this?
How?
No. I'm saying so. Your example: dig up a dead Roman, get his DNA, find two ancestors or descendants from that specific from the Roman Empire, that are living, and then see how many differences have accumulated. Essentially,
and then we can see that it doesn't follow the.
I'm asking you for example, Grayson. What I'm doing. Notice this. Paper after paper supporting everything that I'm saying. So can you cite a paper where they've done this example?
Supporting what you're saying. You're just showing papers saying that hypermutation exists. You're not showing any papers that show this mitochondrial haplogroup has a higher mutation rate. Like these are not supporting like evidences.
Well, firstly, Grits, I don't want you to dodge my question. Can you present a paper?
Okay, I'll give an example.
Okay,
so Jensen, for whom many of these molecular clock arguments originate, he has done this, right? His work has tried to match like known historical migrations of humans to actual like DNA differences, right? Exactly what he's talking about with the DNA differences. And he's been trying to map human history onto the genome going all the way back to Noah, right? So according to his results, right, this is using the same math that Donny is using for these mitochondrial Eve and all that stuff. The same math, he's using the generational mutation rate. He gets that the R1b Y chromosome haplogroup was introduced into Italy in like the 1400s, right? But we know and we have found that members of the Roman Republic were R1b. Okay, so that disqualifies, like that disproves it right there. Like we find ancient DNA evidence that does not fit these predictions. It shows the substitution rate, not the pedigree rate.
Okay, so my response would be a couple things. Okay, firstly, we know that it sounds like you're just hand waving away the evidence that we have for hypermutation. We know it exists today, and we know it exists today for environmental reasons. Going back in time, especially during periods of time where you have harsher environmental conditions, especially if you're living closer to the equator, okay, like a lot of these African populations, and you have mutations to important DNA repair enzymes, which is a thing. Notice here, our findings imply that defects in DNA repair genes can increase germline mutation rates. Okay, so when you look at ancient DNA, or even if you were to look to Neanderthals, okay, one of the most inbred, if not the most inbred people group that has ever listed. Their rungs of homozygosity are massive. They got these stretches of identical letters in their DNA, and they existed in just the harshest of environmental conditions. And they existed isolated in small populations, which means you'd have rapid fixation of new mutations. So if you're looking to the fixation rate, a couple things I want to say here, because you are making some good points. So this is probably the most fun I've had on this specific topic, because most people usually just say they got to look into it and move on. So this is good. But the problem is, although many mutations are lost, okay, like for example, if I were to pass on my Y chromosomal mutations to my sons, it's uniparentally inherited. It's passed on the father's side. Or my wife were to pass on her mitochondrial DNA mutations to my daughter, and then my daughter were to not have children, and my sons were to not have children, well, my question to you, are those mutations passed on then, or do they basically die there?
I mean, if your children didn't have any children, then they're done. They're not in.
Exactly. So my point is, we agree that a lot of mutations are lost. But here's the thing, a lot of mutations are not lost, because there are countless people on the globe that are passing on 100 new mutations per person per generation in your autosomal DNA. They're passing on roughly 3 Y chromosomal mutations per generation, passing on roughly 1 every other generation in the mitochondrial DNA. So eventually there's an equilibrium point where one could say, and this seems to be basic population genetics, that your observed mutation rate eventually equals a fixation rate. Because yes, many are lost, but many are not lost, and many are actually neutral. So the only ones that you're going to lose are the ones where, yeah, if there's no reproduction, there's no passing on of the mutations. Okay, but if you have a mutation that's big enough in, let's say, the mitochondrial DNA or Y chromosome where it kills the organism, then yes, selection removes that individual and that mutation from the equation. But since most mutations are nearly neutral and they don't have a big enough effect to kill the individual, they do get passed on and they do spread and they eventually reach fixation.
Okay,
so go ahead. How do you respond
to it? I think we would both agree, right, with genetic entropy, the odds of any one mutation, right? Every mutation has to either go to fixation or be erased, right? That's the ultimate fate for every mutation, unless that there's a pressure for heterozygosity or there's more mutations that happen at that spot, creating more alleles or whatever. But barring those two things, every single mutation either has to go completely to fixation, 100% of the population has it, or it has to die out. Those are the only two options. This is like basic genetic, like population genetics. So if you're thinking about how many mutations do I have that are inherited from my great-great-great-great-great-great-grandfather or whatever, it's not going to be very many. Most of the mutations that happened in his generation are all died out already after this many generations. Most of my generations are going to be coming from most of my mutations are going to be coming from my generation.
Yeah, if your great grandparents are having kids, and then their kids are having kids, and their kids are having multiple, and it's this consistent passing on of mutations. Yeah. Then no, you have a lot of mutations floating around. Okay, but
only from the recent ancestors.
Are you saying at some point magically you go back thousands of years, and now selection is just so effective that
it's not selection, it's genetic drift. Totally different from selection.
Right. Genetic drift, bottleneck, selection. There's a number of
evolutionary not bottleneck, not selection. It's genetic drift that wipes out 99.9% of all the mutations.
Right. But a lot of PhDs, they'll look to, like Dr. Dan Stern Cardinal, Stefan Frello, who I've debated, they'll look to a variety of mechanisms that can reduce variation. Bottlenecks is one of them.
Sanford and Roop and Carter, the creationists, agree. I've read their paper. They say 99.9% of all mutations are lost because of genetic drift.
But again, as most mutations are lost, there's still a lot of mutations that are not lost.
Yeah.
Because when you're consistently having children and there's a lot of mutations floating around a population that is, and this is where it brings me to population size. Okay. Now, why I would say, I think this comes down to, in our final minutes here before we move on, I don't want to bore the audience. We've been on this for a while. We can leave it up to the audience. Okay. First thing I want to say is I am arguing, and we can leave it up to the audience, your differing size branches in your various haplogroups, whether it's the Y chromosome or the mitochondrial DNA, tells us empirically that mutation rates are variable. You could have some people picking up more mutations in the same amount of time than other people, than their cousins. Okay. So right there, you can't now look at the branches and assume it's X amount of time, because no, if you can pick up more mutations than another people group, or some people groups could be hypermutating for a number of reasons, maybe environment, depending on where you live on the earth. Again, that's an assumption that's a problem for the evolutionary molecular clock. Okay. So, and then the last thing I want to say, and then we'll move on from here, is when it comes to population size. Okay. The biblical model would suggest that at creation, you have two people, Adam and Eve. And then you have rapid and exponential population growth. Then a couple thousand years later, you have a bottleneck. Boom. Okay, so you have a bottleneck, reduces the population to 8. Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and their 3 wives. From there, everybody else is descended from, basically. Those are our ancestors, those 3 reproducing couples. What that means is you have 8 people followed by rapid and exponential population growth, and they don't spread out around the globe until a few hundred years later at the Tower of Babel. What does that mean? You have rapid fixation of new mutations. So you have a faster fixation rate. So during those few hundred years before they spread out at Babel, what do you have? You have a fast mutation rate. You have a rapidly growing population that's basically isolated to a certain area, a geographic location on the globe. That's rapid fixation of new mutations. Then they spread out to all parts of the globe. That's where we find, allele frequency. Some are rare, some are not. If they're rare, then they're more recent. If they're common, they're probably created, okay? You can also find geographically specific Y chromosomes, which is fascinating evidence for the Babel dispersal. But here's the last thing I want to say, and then you can respond. That means with only on average 21 mitochondrial mutations separating any 2 people from the Eve consensus sequence as well, me about 20 mutations from the Eve consensus sequence, that can almost be entirely explained in that first 500 years after the flood, where you have rapid fixation and new mutations. So my point is, what's the most parsimonious explanation given in light of this data? Fast mutation rate, low variation, rapid fixation events after the flood? The biblical creation model. We have no problem with it. But you guys, you got to basically say that purifying selection, has been working overdrive, and the reason why we have such low variation that fits within the biblical time scale, but actually goes back 200,000 years to an out of Africa event, it's because XYZ. And then also, I don't see any prediction. So I just find that the most parsimonious explanation is the separate ancestry model. And go ahead, Grayson.
Hold on. Hold that thought, Grayson. Don't lose your train of thought. Definitely want you to respond to that. Just want to let you guys know we're a bit past the halfway mark. We're loving the conversation. We're looking forward to round 2. But I'm just letting you know that we are past the halfway mark at this point. So if we want to change the subject after you respond to this, Grayson, I suppose you can do that.
Why don't we do this? We'll give Grayson the last word on this round, and then whatever the next round is, I'll get the last word on that one.
Sounds good.
Okay. Sure. Yeah, I'll just say real quick, nobody's arguing that hypermutation happens. Hypermutation definitely happens. It hasn't been shown to happen between large groups of people. That's what that chart was showing, not individuals, but groups of people. You can find one little location on the genome that, this has a higher mutation rate in Africans than Europeans. But you don't see, as a whole, the Y chromosome, the mitochondria, the genome as a whole. You don't see vast differences in mutation rates between people, especially not in the mitochondria. This has never been observed. So until it is observed, I'm just going to say, where's the evidence? And finally, we can make predictions using Jeanson's model, using the molecular clocks of creationists. We can make predictions for what kind of ancient DNA we're going to dig up. And then when we go and dig up the ancient DNA, the whole field of ancient DNA, which, like I said, emerged in the 2010s, it's relatively recently, but we have a lot of material, like thousands of ancient genomes. They debunk this thoroughly. We see the mutations that they have in relation to the consensus sequence that the creationists posit. We see that they don't fit their model. Ancient DNA debunks this. And here finally Donny gets back to the topic. One of the original questions I asked him at the very start of this debate about created heterozygosity. So let's listen to what he has to say about that one, shall we? Because this man really does not understand how alleles and traits work.
An allele is a variant of a gene at a specific loci. There's millions of locations in the genome, and you have millions of spots within the genome where you have capital A, lowercase a, capital B, lowercase b. That is essentially an unlimited potential for variation, chromosomal combinations. Yeah, it's a fact. You're thinking there's just 2 alleles and then that's it. there's genetic variants at specific locations with millions of locations in the genome, and therefore millions of locations in the genome that have the potential for variation. So I don't see it being an issue. You do,
for one trait, that gene is going to be located at one part of the genome, right? So if you're saying that they had like other alleles for the same trait at other places in the genome, we would see the evidence of that. Not everybody would have a gene in the same location on their genome. We would see evidence of for people inheriting like a different allele from a different genome, and that gene would be located at a different loci in their genome if that was the case. And that's not what we see. So it's got to be, 2 alleles per person. That's all you kind of got. I mean, we don't see that there are evidence that alleles
within a single gene,
locations.
Within a single gene, how many single nucleotide variants can you have?
You can have 2 alleles per individual.
But within one gene that spans, let's say, thousands of nucleotides, how many SNVs can you have within that gene?
Are you talking about for a population of people? Like if there were 1,000 people, each one of them could theoretically have a different allele.
Right.
But if they're just one person, they can only have 2 alleles. That's the max for one individual.
Okay, so what I'm going to say is this, and then we can move on, because I feel like we're just going in a circle here. Okay, I get what you're saying, but when you consider the fact that an allele is a variant of a gene at a specific loci, and you have millions of locations in the genome, that means you have millions of spots where you can have capital A, lowercase a, capital B, lowercase b, which just means a state of heterozygosity. If it's capital A, capital B, there's no variation there. It's homozygous, okay? But we also have, from creation, just build a basic family tree, and you can have branches being laid down, lots of time for mutations in different lineages, different groups. We know there's hypermutable regions, okay? Especially the immune system. Yeah, we have a lot of different genes there. It's variable, highly variable, but we also know from the literature that it's highly mutable. There's certain regions that are highly mutable. I mean, how does evolution explain that? That looks like it's designed to mutate in order to adapt to various environments. It looks like we're actually going towards epigenetics, where we can have the environment essentially dictating the organism that's in that respective environment, allowing it to have genes turned on or off for rapid adaptation to that environment. I mean, to me, this screams of forward thinking. Okay, but I know we should move on here. Go ahead if you want to say something quick, and then I'd like to for sure move on to genetic entropy. Go ahead.
No, you can move on. Like I said, I feel like we would just be repeating ourselves at this point, but like if you're thinking about Adam and you're saying he had these 2 alleles at this specific loci, but then for the same trait, he had 2 other alleles at a different location in the genome, right? If you're making that argument, then the populations that we see today should be a mix of having those genes at those 2 different locations. That's what we would expect.
But if you have Adam and Eve that are, let's say, a middle brown skin tone, and so they're capital A, lowercase a, capital B, lowercase b. From that, those DNA differences, those simple DNA differences, okay, you can get every single shade of skin. So yeah, just from that variation there on that one trait, skin color, okay, although there's a lot of other genes that are involved in skin color too. It's not as basic as that, but let's just say for simplification's sake, yeah, that middle brown skin based on the pre-existing heterozygosity, you get every single shade of skin because you have a lot of different mixes there. You have a lot of potential there for variation. You guys, as the evolutionists, you guys are the ones that want to say, well, variations and speciation events, that takes millions of years, because you guys explain the majority of genetic variation as being the result of mutations over time. So you need thousands to millions of years to accumulate mutations, accumulate the necessary variation for selection to act upon and for recombination to reshuffle per generation. But if those differences are already built in from creation, focus on one trait. Sure, take skin color. Capital A, lowercase a, capital B, lowercase b, boom. You have the potential for every single shade of skin on the planet. But that's just one trait. And we have millions of positions in the genome. We have millions of spots that can be heterozygous and therefore tons of variation with all types of traits. So I don't see there being a problem. I guess you do. Have a quick final word, I guess, and then we'll move on to genetic entropy.
My final word on this is that each individual can only ever have 2 alleles, one for each copy of their chromosome, right? It can't be at a different loci in the genome, or we would see that. We don't see that when we look at people's genomes today. Everybody's got them at the same loci, majoritively. Like not, there's non-homologous recombinations and all that, but for like the vast majority of people have them at the same loci. It's not what we'd expect to see. So Adam and Eve would each have a maximum of 2 alleles each, which is not sufficient to explain all the traits. That's my closing remarks. Do you want to skip to junk DNA, or you want to go through? Because like I said, we already talked about genetic entropy a lot in our last debates. If you've got new points to bring up, we can address those. But
good question. I think junk DNA and genetic entropy go hand in hand. Actually, created heterozygosity, what we were just talking about, goes hand in hand too, because the prediction of design diversity, okay, is DNA function. But also with genetic entropy, if the majority of the genome is junk, okay, as you're going to argue, rather than functional, then most of these mutations that accumulate, they can be absorbed by the junk regions of the genome, the neutral regions, and they can essentially absorb the deleterious effects. And so there's no deleterious effects. But if the majority of the genome is functional, let's say between 60 and 80%, that means at least 60 to 80 of those 100 new mutations per person per generation that are accumulating, that means 60 to 80 of those are deleterious. Well, there's been evolutionary population geneticists that have pointed out that even 1 to 3 per generation could be too much. So yeah, I think they go hand in hand, Grayson. So why don't we tackle genetic entropy and junk DNA almost simultaneously, and maybe run with that for the next little bit.
Okay, finally getting to some of the more meatier topics. Genetic entropy. Let's go.
So genetic entropy, what is it? It is the systematic breakdown of the internal biological information systems that make life alive. It results from genetic mutations. Here's an example of one mutation. There's different types of mutations: point mutation, inversion mutation, frame shift mutation, duplication mutation. There's a ton, okay? This is one example. You have a point mutation. Here's the original sequence, TAACTA. Now you get a point mutation, TAACC, okay? So T to C, basically. So mutations accumulate over time. The problem is, and this is where I want to get to this source that I have for you. Okay, so this is from Michael Lynch, Keatley. These are not creationists. These are well-known evolutionary population geneticists and evolutionary biologists. This is from the paper Toward a Realistic Model of Mutations Affecting Fitness. So what do they say? In summary, the vast majority of mutations are deleterious. This is one of the most well-established. So right off the bat, let's not deny that most mutations are deleterious. Okay. The solutions have to take into account this basic fact.
I do deny that.
Well, then you're denying a well-established principle of evolutionary genetics, because they're saying that this is supported by both molecular and quantitative genetic data. So is it your position that you disagree with this summary here?
The consensus of geneticists is the nearly neutral model.
Nearly neutral means slightly deleterious.
No, nearly neutral means that slightly deleterious and slightly beneficial, and all the mutations in between act as nearly neutral mutations.
Beneficial mutations are rare. The only ones that are going to be removed are the high impact mutations. So the ones that are, big enough for selection to see, the ones that are going to kill a person. A single point mutation can kill somebody. Okay, so that's high impact enough for selection to see and remove from the equation.
No, we agree on that. I'm saying the vast majority of mutations are not visible to selection and behave as if they are neutral, because they're within a range where selection doesn't apply to them. Basically, like they're not. It's too minute of a difference for selection to act on.
That's somewhat right. Okay, it's right in the sense that they act as neutral, mutations, as in selection can't see them, they go undetected, they're invisible to selection, okay? Where you're wrong on is the fact that although selection can't see them, they are slightly deleterious in the same way that a single spelling mistake in a book the size of an encyclopedia is deleterious, okay? On their own, these single spelling mistakes, they're inconsequential. You're not going to notice them. But as they accumulate over time, the book, the size of an encyclopedia, it descends into gibberish. The message is lost, okay? It's also akin to rust on a car. Single rust molecules over time on a car, inconsequential, barely noticeable. But after 10 years go by, you need a new car. The car has been
Here's the
destroyed.
difference is that genetic drift gets rid of 99.99% of all that rust after a couple generations.
So that's wrong as well. Okay, so nearly neutral, these low impact deleterious mutations, because they're unselectable, selection can't see them, what they do is they actually spread throughout the population. If selection can't see them, how is selection removing them? How are they removed from the equation?
Selection's not removing them. Genetic drift is.
How is genetic drift removing them from the equation? If they're subject to genetic drift because they're nearly neutral, tell me, how is that solving your problem?
Right. Even your own sources like Sanford, Carter, Roop, they all agree with this. In the creationist model, genetic drift gets rid of 99.99% of mutations. Like, we can pull up the paper. It's their paper on Haldane's ratchet. They say it in the paper themselves. There's just a video last week that Rob Carter put out where he reiterates that fact that 99.99% of mutations.
But he also pointed out that a lot of mutations are not, and that eventually your per generation rate equals the fixation rate in a large population. We exist in a large population. So I need you to expand. I don't know if you even understand what you're saying in terms of genetic drift. If these mutate, okay, let's say people all around the globe, let's make it easy for the audience. People all around the globe are accumulating roughly 100 new mutations per person. They're accumulating those from their parents, from their grandparents, 200 mutations, okay? And it goes back. Every generation is more mutant than the generation before. This is happening population-wide, okay? So how in the world, if these are accumulating, selection can't see them, explain in layman's terms, how is genetic entropy getting rid of these mutations that are accumulating population-wide, spreading through the population as people intermarry and intermingle? How are they being removed? Explain in layperson's terms.
Okay, so you have only 50% of the mutations that each of your parents have, okay? Now, I know Rob Carter wants to point out that those add to 100%. Yes. So the key, though, is that the specific mutations are halved every generation that you're getting through each parental line. So if you go back, like I said earlier, your great-great-great-great-great-great-grandfather or whatever, you are getting almost none of the mutations that he got during his generation. You're saying, oh, but a lot do. That a lot do is like the 0.001% that actually achieve fixation. Because like I said, it's a basic principle in population genetics that every single mutation that happens is bound to either go to reach 100% fixation, which is not very likely, or it's going to be erased in genetic drift, which is very, very likely, over 99%. So the amount of mutations, my genetic load is a sum of all of the genetics of my ancestors, but they began dropping off by 50% every generation. So the odds that I have mutations that many generations ago first impacted my family line are very little. Every generation is wiping these out due to genetic drift. So even though there's no purifying selection, drift is more than capable of wiping out these mutations, like because of sexual reproduction, meiosis, recombination, crossing over, these things. That's the main point.
Okay, so you said this last time, sexual reproduction, meiosis, recombination, genetic drift. No, they don't solve the problems. This is Dr. John Sanford. Here's a source. Highly recommend it. I've got it uploaded on my channel, lecture at the National Institute for Health. Okay, that's not a creationist organization. Genetic entropy, can genome degeneration be stopped, degradation be stopped? It's a legit problem, and he discusses throughout this lecture that these mechanisms have been looked at. Okay, contrary to what the critics say about Mendel's accountant, results from Mendel's accountant, which utilizes realistic parameters. You can change it. You can say this many beneficials or this little of beneficials, this much junk, this little junk, this amount of selection. You can utilize realistic parameters. They've actually published in secular journals, okay, the results of these numerical simulations. And the results are always the same, that degeneration is inevitable. Okay, what you're saying doesn't work because, again, you have mutations population wide. Let's just say, and I said this the last time, but I think it's an irrefutable argument, so I'm going to say it again. Okay, we can look to the recombination. We can look to some form of purifying selection. We can look to beneficial mutations. Let's just say that 8 billion people on the planet, selection, you remove 50% of the worst mutants. We're all multiply mutant. Every generation is more mutant than the previous generation. That's the point. Okay. Regardless of everything you're saying here, you still have every generation being more mutant than the prior generation. That is a fact. You can't get away with that. Okay.
That's what I'm trying to address.
Even
is that, like, in Mendel's accountant, in the papers that Sanford is publishing, they say 99.99% of every mutation is wiped out from genetic drift. That's in these papers. That's in Mendel's accountant. Like, if you have enough generations here, like, there's a huge tapering off effect. You get an asymptotic approach to zero as you start factoring in which mutations you're inheriting from your distant ancestors, right? So as you go along, you're at a genetic load homeostasis, right? Because every generation is getting, there's a die-off effect of mutations that you're inheriting from your deep ancestors, and every generation that, like, tracks forward. So there's not an increase in the total mutations, like, and the total genetic load per generation. That's
yes,
not
there is. Just wait, because you butted in, I lost my train of thought. I just want to finish the point I'm making. Okay. The problem is, even if you were to invoke unrealistic amounts of purifying selection and you got rid of 50% of the worst mutants on the planet, you're left with 4 billion. You're still left with 4 billion people that are more mutant than the previous generation. If you invoke too much selection, then that results in extinction. So you can't, selection's limited. Plus, there's a lot of noise going on, okay? Because you have a lot of nearly neutral mutations that are accumulating. You have your rare beneficial mutations accumulating, okay, although most of them aren't even selected and manifested, simply because of the noise that's going on. And what you're saying, in terms, you're misrepresenting what Dr. Carter and Dr. Sanford are pointing out, because they themselves say that no matter what you look to, no matter what parameters you use, for example, here, mutations are realistic parameters. Mutations are overwhelmingly deleterious. I actually confirmed this with Dr. Sanford himself. Okay. Most mutations are subtle, so they get lost in the noise. They are accumulated unchecked. They go on. They don't have to reach fixation. Not every single nearly neutral, low impact deleterious mutation needs to be reach fixation. They just need to accumulate in the population. They result in disease. That's why every single year you have thousands of new mutation related disease popping up. In his lecture at the National Institute for Health, Dr. Sanford pointed out that autoimmune diseases are on the rise. Cancer is like 1 in 3. Autism might have a genetic component to it. There's immunological issues due to mutations. Mutations are causing problems. And every single time they invoke realistic parameters in these numerical simulations, even when you assume 90% of the genome is junk, the simulations manifest results that say that genetic degeneration is inevitable. Populations descend into sickness. They don't have to reach fixation. They just have to float around the population. They result more and more in degeneration. Okay, we've seen small scale examples of genetic degeneration with Neanderthals, with mammoths, with isolated populations like butterflies, with the Hobbit so-called pre-human ancestor. So no, I don't think your answers are cutting it. I don't think you're dealing with the problem. Go ahead, Grayson.
Right. Yeah. The key in those examples is that there's small, isolated populations that are dying out because of environmental reasons. The mammoth, Neanderthal, all of these things died out not because they reached genetic entropy and like they died out because they couldn't cut it because the environment changed in the case of the mammoth, or overhunting, or they got out competed by like Neanderthals did. There are other reasons that explain why these species went extinct. It's not genetic entropy, which doesn't exist. It's not a thing. Like when we look, like the prediction of genetic entropy is that every generation should have a higher gene load, like a mutation load, than the previous. This is very easy to empirically verify. We can just go to the lab, get a sample, measure the genetic load per generation, and we see it's the same. It doesn't increase every generation.
No, that's wrong. That's wrong. Okay, so you made a few points here that are wrong. Okay, so firstly, I want to point out that your population geneticists that are not even creationists, okay, they agree, for example, Lynch and Crow, that humans are degenerating at a rate of between 1 to 5% of fitness per each generation. So yeah, each generation we are getting more and more degenerate,
we know
more
why
and
that
more
is.
sick, more and more unhealthy. These numerical simulations have demonstrated that. These examples in mammoths and Neanderthals, yes, they're isolated, they're inbred. What that means is your homozygous alleles that are accumulating, you're getting more homozygous spots in the genome, less heterozygous spots, and you have more deleterious mutations that could lead to harm because they're being manifested. You got this hidden reservoir of genetic mistakes that are now manifested. They lead to rapid and accelerated genetic degeneration. That's microgenetic entropy. But in the human population, we have 8 billion people. The more mutations that accumulate generation after generation, the more homozygous alleles there are, less heterozygous alleles. And basically, you eventually descend into something similar that these small, isolated, inbred populations descend into. It takes longer for large populations to descend into that. But again, these famous geneticists are pointing out that humans are degenerating more and more every single year. Every chromosome, as you can see here, has a complex array of nearly neutral and deleterious mutations. That means every generation is going to be more and more mutant. And again, I don't feel like you're adequately dealing with it.
Right.
I feel like you're disagreeing with these population geneticists. Go ahead.
Genetic drift totally deals with this, but beyond the point.
No.
Yeah,
all you're saying is genetic drift.
Wait. As I was saying, with mammoths, Neanderthals, and humans, there are environmental reasons that we would observe the genomes that we do, right? Like with humans, as I said, there's relaxed selection, medical technology that is clearly the driver of this. Even Lynch says in his paper that, like, we observe this effect in industrialized nations that have access to medical technology. We're not seeing this in hunter-gatherers. I mean,
all right. Hey, so that's time.
And of course, the final subject is junk DNA, because creationists know that if they acknowledge the existence of junk DNA, then their position is instantly defeated, because junk DNA is not sequence constrained. That's just a fact. That's an observable fact of the world. And yet, the sequences of junk DNA reflect the same nested hierarchical pattern of genetic homologies that we would expect and do in fact observe in the theory of evolution and in reality. And this is why they have to cling so hard to the idea that the genome is 100% functional, because if they admit otherwise, then they also admit that evolution is a fact, and they just can't have that, can they? Junk DNA, pretty easy to understand. Most of the DNA doesn't have a known function. Shouldn't have a known function. We can measure how many of the sequences are constrained versus unconstrained, meaning that it doesn't matter what letter of the G of ACTG it is if it's unconstrained. We get measurements, 91% unconstrained, 9% constrained. We can measure based on mutation rates. We get the same, like 2 to 10% of the genome should be functional based on the mutation rate that we observe. But the main argument that I like the best for junk DNA is the observation that there is no correlation between complexity of an organism and the size of its genome, like the amount of DNA that it has. So this is what I've been waiting to ask, is called the onion question. So we know that humans have about 3 billion base pairs, and onions have about 16 billion base pairs. So the question for Donny is that if all of this DNA is truly functional and it's not junk, do you believe that you are less than 4 times as complex as an onion? Right? Is an onion that much more complicated than a human, made in the image of God, that God requires 4 to 5 times more DNA to make an onion than a human? So that's just where I'll leave it for the start.
Okay. I appreciate it, Grayson. I wrote that down. So let me start from the top and I will get to your onion question. Good question. And this is a good topic to now dive into after the topic of genetic entropy, because essentially if the majority of the genome is functional, then that's strong evidence for created heterozygosity, because that's a direct prediction of the design diversity model that suggests that the majority of DNA elements, including pseudogenes, endogenous retroviruses, and allele sequences, are functional, beneficial to the respective organism. It also refutes evolution and basically props up genetic entropy, because now more mutations are accumulating that are deleterious, and so those can drift throughout the population, float around. I just have to add this in, and once you go next, you can say something, because it does kind of blend. Is genetic entropy and genetic drift actually makes genetic entropy even stronger, because if these functional regions of the genome are hit with mutations, these mutations are unselectable, they're invisible, and so they're subject to genetic drift. They float around. Okay, they're not removed. They float around, resulting in more genetic sickness, and some get fixed. So either the ones that float around or get fixed, it's still bad. It's no help. Okay, but so this is why it's so important, because again, it's a double whammy. DNA function demonstrates 2 of the major creationist models. So in 2012, scientists were surprised to see that, and you can see here, Encyclopedia of DNA Elements, the ENCODE project. They assigned at least 80% of the genome was active. Transcriptions about 60%. Over the years, there's been follow-up papers, and there's debate now. Is it 60%? Is it 80%? Either way, any more than, let's say, 20%, realistically 10% is too much for evolution. Okay, so the biochemical approach, I do have slides here demonstrating why that approach is the best in terms of function, and also why the genome-wide activity really is legit activity and not just noise or spurious, as Grayson would argue here. And here's a couple reasons, then I'll wrap it up with the onion question and then take as long as you want, Grayson. So firstly, I said this in our first debate, transcription, it's an energy intensive resource. It's a resource intensive process, releases a lot of heat, releases a lot of energy, and nucleotides are being utilized literally in this process to produce RNA molecules that, if they were to just end up being noise or spurious, that's problematic, because of the fact that it is an energy intensive process. Every time you add a nucleotide to a growing RNA chain, you're consuming a lot of energy. ATP, which is the main source of energy for the cell, is used in many of the stages of transcription. Okay, DNA to RNA to protein, from RNA to protein's translation. So we're talking about DNA to RNA, these functional RNA transcripts. They function in a variety of ways. So it would be faulty to assume that most of the non-coding region is non-functional evolutionary leftovers, essentially. So again, if this was just noise, if this was just spurious, then and just junk, this unnecessary transcription, and Grayson has some responses to this, so we'll deal with that. It should be dealt with through various evolutionary mechanisms. If that were the case, over time, millions of years, selection should remove this junk, because it's just wasting energy, wasting resources for the cell. Or there should even be mechanisms in place that can suppress this junk and prevent it from being transcribed, and therefore using all that ATP and using all of the energy that's required. So his last question here, the onion question. So that is a very good question, and it comes down to the fact that what we have in the genome, the genomes of living organisms. This includes plants. This includes various species of fish, like lungfish. This includes a lot of your different mammals, or some of your salamanders. There's certain animals and plants, especially, that have 50 times more DNA than humans. Well, why is that the case? Well, firstly, the amount of DNA that an organism has doesn't necessarily tell us how complex that organism is. Okay, just because an organism has more DNA does not mean that it's more complex than, let's say, a human being. Let's say a certain plant has more DNA than the human. That doesn't mean that it's more complex, okay? And we have, in the design diversity model, we have something that is called over-engineering. Okay, and what this is, I'll make it understandable, is when you design or build a bridge, you don't just build a bridge to hold up the weight of the exact number of cars that you expect to drive on the bridge. You build it 10 to 20 times stronger than the number of vehicles that you expect to be driving on the bridge. This is called over-engineering or over-designing. And it is, as a matter of fact, it is good design. This type of design adds robustness and reliability. How does this answer his question? Well, a lot of plants, they are subjected, and especially you can read papers about lungfish and the harsh environmental conditions that are rapidly changing that they exist in. Think about the rapidly changing weather-related conditions that plants exist in. Okay, we're humans, we live in a house, we have shelter, we can put clothes on, we can protect ourselves from harsh environmental conditions. Plants can't do this. Okay, your plants can't put on a coat, build a house, so they need a lot of redundancy. They are over-engineered in order to survive, to have robustness in terms of these ever-changing environments, these harsh environments. And there's evidence for this. We have paper upon paper that shows rapid evolution. They'll say rapid evolution, but it's actually rapid adaptation of various plant species, various fish species. They have these fish species that enter cold water. Okay, that's not the environment they existed in, but because of the pre-existing ability to adapt based on the redundancy, the over-engineering, epigenetics, for example, they actually rapidly adapt to this new environment. Rapid adaptation based on the pre-existing ability to do so. And so that's why you have some plants that actually have 50 times more DNA than humans, is because they are over-engineered. They are designed with a lot of redundancy for survivability, robustness, and adaptation. I said a lot there, Grayson, so take your time.
Okay, so first you said you're talking about the ENCODE results. So I would just point out that ENCODE itself came out with how much each of these regions is being transcribed. So they put out the rates of these transcriptions, and we see that about 70% of that 80% number, so 70% of all of the transcribed RNA that ENCODE measured was at a concentration of like 1 transcript per cell. In biochemistry, you're not going to have any functionality with something that's only prescribed 1 transcript per cell. That's very quickly degraded by the enzymes around it. It's not having an effect. So ENCODE showed that most of this stuff is spurious. And then Donny brought up this energy problem, saying that, it takes so much energy to do all this transcription. I would like to point out the fact that junk DNA is somewhat special for eukaryotic cells. We don't observe this much spurious transcription in prokaryotes. And what do eukaryotes have that prokaryotes do not? They have mitochondria. So mitochondria increase the amount of energy that's available to a eukaryotic cell by nearly 1,000% fold from prokaryotes. I mean, it's like a huge increase in the available DNA, in the available energy that the cell has. And then they also have a nucleus. So all of these transcripts that are produced are contained within the nucleus. So they can't leak out and have all these effects elsewhere. The enzymes within the nucleus quickly break them down. And actually, we see a lot of the computation about saying, which transcripts will make it on to be translated. We see that in a lot of DNA splicing. So when a transcript is spliced and the order is changed up, it inquires certain markers that the cell machinery uses to figure out where it needs to go, like to sort it, basically. And most of these transcripts don't have any spliceosome interaction. They don't have any introns to splice out. There's no splicing activity. They're just created and pop. They're degraded. We can see this in like ERVs, for example, where you look at, for something to be functional, right? For something to be transcribed, it's got to have, or translated, it's got to have codons. It's got to have an open reading frame. So a start codon, a stop codon, everything in between, that's what's read. So only 0.15% of ERVs even have an open reading frame. So they can't, only 0.15% can even be read by the cell's machinery. So it's a very, very, very small percentage of the total ERVs. And then finally, I don't think that really addresses the onion problem, because more DNA, if all the DNA is functional and doing a function, more DNA does have to mean more complex. If there's more functions going on in the genome, in an organism, like that has to be more complex. I mean, you have more things going on, more functionality in the genome that's bigger if all these genes are functional. So I don't think that rescues you. And then I would just point to organisms like tardigrades that have, they're the most robust. They can survive the most different situations of any organism alive. What's their genome size? Is it the largest genome of any organism? No. So it's not like these things correlate to robustness or how these, it doesn't correlate to that either. I mean, domesticated wheat can get knocked out by all kinds of conditions, and yet, it's got a ton of DNA. So these things don't equal robustness either. I mean, it doesn't fit in a paradigm that says that most of the genome is functional. It just doesn't compute.
Okay. Let me just make sure I'm unmuted. All right. Lots there and lots of fun points to address. So where should I start? Okay. I'll start from the beginning then and kind of move from there. So you said that ENCODE themselves essentially revealed that most of this activity and transcription was spurious, was noise, not really doing anything. What's funny is, even for sake of argument, this isn't the case, I'm going to refute it. Okay, but just for sake of argument, even if the genome was only 10 to 20% functional, okay, and this is why genetic entropy and junk DNA goes hand in hand. Okay, what that means is you still have 10 deleterious mutations accumulating from generation to generation, even if the genome is 10% functional. Okay, any more than that, so say it's 20, it's 30, it's 60, then that's 20, 30, and 60 new deleterious mutations accumulating per generation. That makes evolution impossible, essentially, okay? And there have been population geneticists in the past that have pointed out that even one per generation could be an issue. This is why the evolutionary community needs as much junk as possible. Again, I know the evolutionists hate it, but Mendel's accountant, the results of it have manifested in secular journals, and they've demonstrated that a genome of 90% junk and 10% function is still too much for evolution to proceed. Okay, here's just a couple excerpts from papers where we see rapid evolution in plant species. Animals are evolving faster than you think. Again, there's a lot of functional redundancy. There's a lot of over design. Okay, you're not going to build your bridge with in mind, well, only 10 cars are going to drive over this, so I'm going to design the bridge to hold 10 cars. No, you're going to design the bridge to hold hundreds of cars, over design. Now, here's the thing. If you have plants with with a lot of redundancy. Here's an example of the evolutionist just not wanting to tap out. Again, the size of the genome, even if you have a genome that has more redundancy, let's say 30 times more redundancy in plants than in humans, or even just more areas of mutation buffer, that doesn't make it more complex, because humans, being higher mammals, have genetic sequences and DNA elements that are involved in what? The brain activity, heart activity, the circulatory system, the respiratory system. There's so many more complex organs in a higher mammal than in a plant species. Even these endogenous retroviruses he's talking about here, I wrote a whole book on it. And I think Grayson and I should do our third debate only on chromosome 2 fusion and endogenous retroviruses, because this could take another 3 hours. Endogenous retroviruses, there's paper after paper telling us that if we didn't have endogenous retroviruses, we wouldn't exist, because they function in embryological development, they function in the immune system, they function in determining cell types, they function as antiviral protectors. And the reason why they are built the way they are, with your LTRs, long terminal repeats, your ENV, your GAG, your POL genes, okay, they're designed that way because they use a mechanism called viral mimicry, where firstly they can fight off tumors. Okay. And they can also fight off invading exogenous retroviruses. So they need that sequence similarity to exogenous retroviruses to carry out, to systematically carry out many of their functional roles. Okay. These are functions and these are organs that are essentially workable through DNA sequences and DNA elements that onions and plants simply don't have. So just because they have more areas of redundancy and over design doesn't make them more complex. So that simply doesn't work. Okay. So again, I don't find that this argument is working, because low level transcription, that doesn't matter. If firstly, it's like the chromosome 2 fusion. This is why we should talk about it briefly. They try and say, oh, the DDX11L2 or the DDX, 11L2 gene, it's just expressed at low levels transcription. Firstly, it's apples and oranges. When you're comparing protein coding sections of the genome and non-protein coding sections of the genome, your low level transcription in your non-protein coding regions of the genome, that could equate to high levels of function versus a higher or lower transcription in protein coding regions. Okay, that's why they know this, and that's why they look for what's called co-expression. And so we know that the DDX11L2 gene is functional. It's not a pseudogene. It lies within the first intron. You've got Ken Miller that's trying to say that it's actually outside, but it's actually, he's been corrected on that. Anyways, I'm just going on a rabbit trail. This applies to the low level transcription. Who cares if things are transcribed at a low levels? That doesn't mean they're non-functional. There's a lot of ERV elements that are completely turned off. They're involved in embryonic development. We have these ERVs that are involved in, from zygote to baby, fully formed, healthy baby. There's these various developmental windows that these ERVs are functional in, but once they're done their job, they're turned off. So these evolutionists, with their evolutionary assumptions, they assume, well, these are turned off, or we knock them out and they don't affect the organism, then they're junk. No, because a lot are turned off, and they're only, either they've done their job or they're there and waiting to be called upon via environment or epigenetics. Okay. My car, I rarely use the rear wiper blade. I can't remember the last time. I use the front wiper blades all the time. I rarely use the rear wiper blade. Okay, so because I barely use that, and does that mean that it's not functional? No, it's there for when I need it. The spare tire in the back of the car, if I get a flat tire, thank God I got that spare tire, because it's going to come in handy. That's an example of redundancy. The rear windshield wiper could be an example of what they say is transcribed at a low level. Let's just, it doesn't mean non-functional is the point. So again, here's a few other reasons. I should point out though, that in 2017, a research team looking at non-coding RNAs argued that most, I think I've got the papers here. Let's see. Knockout tests reveal function. Notice this. Taken together, these knockout analyses of microRNAs and long non-coding RNA. There's at least twice as many long non-coding RNAs as there are protein coding ones. and they're functional just in a multitude of ways. You know, we have unexpected functional roles of these long non-coding RNAs. But anyways, indicate that non-coding RNAs are certainly important in embryonic development and disease progression. So they're snipped out, they're knocked out, and it results in disease. Okay, so there's paper upon paper about junk DNA essentially being overturned. Okay, but the main argument is this noise argument. So there's two papers that I think are significant in this. And Grace, I know you'll want a response. So just make sure you're writing this stuff down. But you brought up a lot of points. I want to address them all. Okay, so a research team, secular research team, they were looking at non-coding RNAs. And they argued that most transcripts actually play some kind of functional role. They determined that non-coding RNAs are probably not just transcriptional byproducts, but comprise a diverse population of functional RNA transcripts. What's the point? Well, the point is we have a research team that worked independently of the ENCODE project. And Grayson wants to say, well, the ENCODE project, their results suggest that it's mostly spurious and noise. That's not the case. We have an independent team that are concluding that the transcription observed in the genome is mostly functional and not just biochemical noise. So that means this independent research further strengthens the results of the ENCODE project. Okay. And then there was another paper in 2016 where but they didn't. Grayson, as you're going, I'm going to find the papers. I've got hundreds of slides here, but I have them for you up here. So
just to let you know, we're past that 15-minute mark. So if you want to maybe finish this up, and then we'll let Grayson have a few minutes to respond, and then you want to just jump right into the closings.
Yeah. Okay, I'll wrap it up. I'll wrap it up here with this point here. Okay. So again, if these transcripts were just noise and, you know, they weren't functional, then this would just clutter up the interior of the cell if they weren't playing any kind of functional role. But the most important point, and I'll wrap it up with here, is it easier to keep junk around for millions of years? I find Grayson was downplaying how much energy the ATP being used for transcription. I feel like he was downplaying it. Okay, because you could just go through, study the process of itself and everything involved with the RNA polymerase and the promoter and just the process of taking the gene's DNA sequence, transcribing it to make an RNA molecule. Okay, you've got transcription initiation, elongation, transcription termination. The elongation stage requires, notice this requires lots of ATP. Okay, ATP is needed for transcription. For example, at the point where transcription factors get attached to the promoter region of DNA and also in the adding of nucleotides during elongation. Energy is also needed when the stop codon stage is reached and the mRNA, messenger RNA, is detached from the DNA. All steps of transcription require energy. Guys, it's an energy intensive process. And to just say that the genome is just transcribing it, it's easier to do so, rather than, if evolution is true, having selection remove it in order to declutter the system, to say that is just an absolute, it's without evidence. It's an evolutionary derived rescue mechanism. It makes no sense. To say that it's just easier to transcribe it, even though it requires all of this energy, it doesn't make any sense scientifically. So I'll yield there. Grayson, go ahead.
Wow, that was so many points. Okay, I'll try to work and address all of them. I hope I don't miss any of them. But first of all, the kind of energy that we're talking about, not that much. I mean, I don't even know if it amounts to one extra bite of food for an organism. Like, we're talking about on an organism level, how much more energy do you need to consume to support these extra transcription processes? It's not a lot. And furthermore, I think the main point that I'm saying is that most of this is outside of the like what selection sees, right? These energy differences are small enough to where on an organism level, there's other more pressing factors that are determining your overall fitness. So in terms of the overall impact on an organism's fitness that natural selection would see, because of these like spurious transcriptions, so like very 70% of these were like one transcript per cell. So the challenge I have is find one functional area of the genome that only requires one transcript per cell. I don't think you can do it. I don't think that anything that we know that is functional only gets one transcript per cell. That is such a small concentration compared to everything else that we know of that actually plays a functional role and how much it's being transcribed. So you mentioned a lot of ERVs. You mentioned ALUs. You mentioned long non-coding RNA. All of these things, when you hear the functions of these, just keep in mind what percent of these are functional. It's like not even 1% of them are functional. Like that, the function that we actually do know, because most of these categories do have a very, 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 very small function. Like I said, for ERVs, it's not even a tenth of a percent that are shown to be functional. And this is like not just saying, oh, well, we don't know, they could be transcribed. We know the whole transcriptome. Like we know everything that's transcribed. ENCODE showed this and the amounts. And we know that like 1/10 of a percent is even transcribed. And that stuff is just degraded very quickly after it's transcribed in the genome. This kind of thing is not a big impact on fitness. It's not something that natural selection even selects on because there's way more pressing factors. Like there's more pressing mutations that you have that are going to impact your fitness more than needing a little bit more energy to transcribe like one transcript of all of these different areas. I mean, look at the things that actually have function. Look how much they're transcribed. It's like a lot. If you made it this far into the video, thank you so much. I hope you enjoyed listening to that debate. Maybe you learned a couple things about genetics and countering apologetics of creationists that try to actively to misrepresent science. Anyways, guys, that's it for me for now. So hopefully you enjoyed the debate. Like and subscribe if you want to see more debates against creationists, flat earthers, and just overall silly pseudoscientists in general. Remember, stay curious and keep learning.